Good afternoon. Let me begin by thanking the organizing committee for the invitation to speak here today. My laboratory has been involved in drug development for over 3 decades. One of our earliest successes involved the discovery and development of 2 key antiretroviral agents, lamivudine and emtricitabine. These 2 drugs are components of 17 FDA-approved combination therapies for the treatment of AIDS. While each of these combinations play a significant role in slowing the spread of the disease, I'd like to focus on the historical importance of one of them, a triplet. Next slide. One of the first breakthroughs we had in the treatment of AIDS came in the mid-1990s with the development of highly active antiretroviral therapy, oftentimes referred to as HAART. By following HAART protocols, we could, for the first time, dramatically slow down the onset of viral resistance. Unfortunately, in these early regimens, patients were required to take 15 to 20 pills a day, usually in 3 increments, to avoid developing resistant mutants. So that's the panel you see on the left. However, the game changer came in 2006 with the introduction of a triplet, which combines 3 drugs in 1 pill taken once a day. While not a perfect therapy, a triplet dramatically improved patient compliance and allowed patients to lead near normal lives. Emory's Office of Technology Transfer estimates that over 90% of patients in the US on antiretroviral therapy take or have taken one of these drugs discovered at Emory. While this was a wonderful achievement, the bulk of the work that my laboratory did in discovering and developing lamivudine and emtricitabine was done years ago. So for the rest of today's presentation, I'd like to highlight studies that are more current. To do this, I've asked 3 scientists in my group to speak about projects they are leading. Given that this is the BioAfrica conference, I've chosen 3 individuals who did their undergraduate and graduate training in South Africa. The first person you'll be hearing from is Dr. Nicole Privet, who received her PhD degree from the University of Stellenbosch. Her presentation will be followed by one from Dr. Luke Hodson, who also received his PhD degree from the University of Stellenbosch. Subsequently, you'll hear an interesting story from Dr. Stephen Pelley, who received his PhD from WITS. I will then return at the end to tell you about our nonprofit drug development company, Drive, and highlight development of Molnupavir, a clinical stage drug for treating COVID-19 infections. I hope you find our story to be interesting. Nicole, the floor is yours.
Thank you. Okay, so I will be giving a talk on improving the pharmacokinetic properties of tenofovir prodrugs, tenofovir being one of the components of a triplet, which Dr. Liotta spoke about in the previous slide. Steve, if you would move forward. So tenofovir is an acyclic nucleotide phosphonate. Again, it's one of the cornerstone prophylactic and therapeutic approaches to controlling HIV. It's present in almost all combination antiretroviral regimens. Tenofovir exerts its therapeutic effect after in vivo conversion to the mono and then active diphosphate species shown at the bottom right. It competes with endogenous nucleosides for incorporation by viral reverse transcriptase, where it then prevents further propagation of viral RNA into viral DNA. Now, the advantages of tenofovir is that it has this intrinsic phosphonate, bottom left, highlighted in blue, which is not present for on more natural nucleoside antiretrovirals such as emtricitabine shown just above it. And the advantage to this is that it bypasses that first phosphorylation step, which is typically the rate limiting step, and is also more metabolically stable than the corresponding phosphate, due to the fact that it has this phosphonate linkage, which is resistant to phosphodiesterase cleavage. One of the drawbacks, however, is because of this intrinsic phosphonate, tenofovir is negatively charged at physiological pH. And this means that it has very limited oral bioavailability and it struggles to cross cellular membranes in order to exert its antiretroviral effects. And furthermore, because it tends to then concentrate in plasma and then in the kidneys, it can cause associated toxicities such as nephrotoxicity, and is also been known to be responsible for bone mineral density depletion. Next slide. We can overcome this problem by formulating tenofovir as a prodrug. And in fact, there are 2 tenofovir prodrugs that are approved and form essentially the cornerstone of most combination antiretroviral treatments. And this is tenofovir disoproxil fumarate, or TDF, and tenofovir alafenamide. And both these prodrugs are able to successfully mask that negatively charged phosphonate in order to increase cellular uptake and increase the efficacy of tenofovir and its antiviral properties. However, while they're able to do this, they're not without their own disadvantages. TDF, for example, is non-specific cleaved by plasma and hepatic esterases, which means that you get premature release of tenofovir in the liver and in plasma, which can lead to those organ-specific toxicities that I've mentioned earlier. Now, TAF, on the other hand, was developed to overcome these toxicity issues with TDF by remaining stable in plasma and targeting an intracellular cleavage mechanism by cathepsin A to release tenofovir within cells. However, tenofovir alafenamide is actually largely extracted by the liver due to cleavage by carboxylesterase 1. And so what you end up getting is about 65% of TAF being cleaved by the liver, which means that only a small percentage of prodrug is then available to enter into systemic circulation and access HIV-infected cells. Next slide. Another prodrug of tenofovir which has been developed is this tenofovir exaladex, which disguises the acyclic nucleoside phosphonate as a lysophospholipid analog in order to increase its oral bioavailability and cellular permeability by utilizing natural lipid uptake pathways. And this, of course, can lead to enhanced cellular uptake, increased intracellular levels of tenofovir diphosphate, and can significantly improve the efficacy of these prodrugs relative to tenofovir. And tenofovir exaladex, like TAF, next slide, was also designed to be stable in plasma to reduce those toxicities associated with tenofovir itself and actually TDF. And this was achieved by replacing the acyl ester linkages of normal phosphatidylcholine derivatives with these ether and proton. And this is to remove potential cleavage by phospholipase A1 and A2 upon absorption. What's more is, as I mentioned previously, because of the phosphonate, you get that phosphonate linkage instead of the phosphate, and so you reduce the potential for phosphodiesterase cleavage, which leaves only cleavage by phospholipase C, an enzyme which is not found in plasma, but only found nested in the inner leaflet of the plasma membrane. And so once again, you're targeting only an intracellular cleavage mechanism to release tenofovir. However, like TAF, TXL is highly extracted by the liver, and this is due to the CYP450-mediated omega oxidation, which introduces this hydroxyl group at the terminus of the lipid chain. This then undergoes a series of oxidations, which can then degrade the lipid 2 carbon atoms at a time to ultimately release a large proportion of tenofovir within the liver. And so once again, you're delivering only a small fraction of intact prodrug to HIV-infected cells. Next slide. Now, our group thought we could overcome this issue by introducing metabolically stable motifs at the terminus of the lipid chain. This will prevent that CYP450-mediated omega oxidation and thereby reduce toxicities associated with premature cleavage in the liver and plasma, extend the circulating half-life of these prodrugs by extending their duration of action, and of course, delivering a much larger fraction of intact prodrug to HIV-infected cells, which could essentially reduce the amount of prodrug needed in order to have the same kind of efficacy. because you're increasing the intracellular levels of tenofovir diphosphate. And so we ran a series of SAR, and the idea was to get prodrugs that would not only have the same efficacy as TXL, as shown here, which was about 9 nanomolar in vitro assay, but also significantly improve metabolic stability, at least as a first pass in human liver microsomes. So for TXL was 42 minutes, and we wanted to get prodrugs with significantly better half-lives. So if you go to the next slide. So to this end, we came across a large number of lipid prodrugs featuring terminal motifs that either through electronic effects or steric effects were able to mitigate that androgen omega oxidation at the terminus of the lipid chain. And you can see this is just a select example of compounds which not only showed the same sort of potency as tenofovir Xaladex, but also demonstrated significantly enhanced stability in human liver microsomes with half-lives of greater than 120 minutes. And so we wanted to see now we have these amazing properties that in an in vitro setting, would this translate to an in vivo setting? And so we selected 3 of these compounds to test in an in vivo mouse model to see if we would see the same sort of stability in mouse liver. And so we took the 3 and administered a single dose orally to mice, and then quantified the amount of prodrug and tenofovir as a metabolite in liver and plasma samples. And if you go to the next slide, and here we see the results of the mouse plasma PK experiment. And so you can see across the board that our prodrugs have achieved an elevated systemic exposure levels in plasma relative to TXL, which is indicated by the area under the curve. What's more is compound 158 in particular far outperformed the others. Furthermore, we can see that compounds 130 and 158 also had enhanced relative levels of prodrug relative to tenofovir as the metabolite, which reflects our goal to reduce plasma tenofovir levels, thereby decreasing associated toxicities and increasing the amount of prodrug in plasma, which is intact and can then access HIV-infected cells. Importantly, compound 158 exhibited an improved half-life of 8.66 hours, a 3-fold improvement relative to TXL. We know that TXL has been developed or was being developed as a once-weekly antiretroviral, and we have shown a longer circulating half-life relative to TXL, which means that prodrugs such as compound 158 have the potential to enable dosing regimens that could exceed once a week. Next slide. We were also able to demonstrate that these promising pharmacokinetic properties were also observed within the liver. Once again, we can see that compounds 130 and 158 showed enhanced exposure levels in liver relative to TXL, and the levels of prodrug relative to tenofovir, the metabolite, were also enhanced for these compounds, demonstrating the utility of these omega functionalized prodrugs to mitigate metabolism within the liver. And so we have developed this proof of concept, which can show that our prodrugs offer safety and efficacy advantages over current tenofovir prodrugs. And what we have shown here is the benefit of these prodrugs in the context of HIV. However, next slide. The real takeaway here is that we can show that the prodrugs developed in the Liotta group serve as a platform which could be applied to improve the efficacy, safety, pharmacokinetic properties, and ultimately the long-acting potential of antiretrovirals targeting a variety of other diseases for which no such treatment options exist. And it is at this point that I'm going to pass the baton over to Dr. Luke Hodson, who's going to describe another avenue of this prodrug platform and how it has been applied to treat neurological diseases.
All right, thanks, Nicole. So as Nicole showed in her talk, we're very interested in utilizing prodrug strategies within the Liotta research group. And for my part of the talk, I'm just going to be speaking to you about a project that I'm working on, which is developing prodrugs of cannabidiol or CBD for the treatment of refractive epilepsies. Next slide, Steve. So roughly a third of epilepsy patients do not respond to anti-seizure medication, despite the introduction of numerous new medications over the last 3 decades. And this problem is illustrated within the graph shown on the left over here. where the patient response rate to various therapies over the last 3 decades is plotted over an extended treatment duration. So you can see for the 1980s, the patient response is plotted in blue, whereas in the 1990s, it's in red, and in the early 2000s, it's shown in green. And what you can clearly take away from this graph is that roughly the graph plateaus at around 2/3. And what this means is that a third of these patients, which equates to roughly 1 million people within the USA, are refractory to these existing medications and suffer from inadequate seizure control. And if you think of it in the context that 80% of the global epilepsy population resides in low to middle income countries, and around 5.4 million people are affected in sub-Saharan Africa, the numbers of refractive patients are much higher in these areas, and this is truly a global problem. So this disappointing lack of progress is mainly due to the fact that virtually all of the new medications that are being developed target the same mechanisms as these existing anticonvulsant medications. So this is why we haven't been able to treat these refractive patient populations and make a dent in this. Next slide, Steve. So cannabidiol, or CBD, which is a non-psychogenic component of cannabis, was recently demonstrated to be an effective anticonvulsant treatment for these refractive epilepsies. So CBD was approved by the FDA as Epidiolex in 2018 for Dravet and Lennox-Gastaut syndromes, which are both true forms of epilepsy, as well as for tuberous sclerosis complex. And while Epidiolex is effective, it's not necessarily that well tolerated in its patient population. But Epidiolex has achieved a large amount of success with an annual revenue of around $500 million in 2020. And the parent company, GW Pharma, was acquired by Jazz Pharmaceuticals for over $7 billion in March of this year. So the success that CBD finds in treating these epilepsy patients can most certainly be attributed to the fact that CBD acts on multiple different targets, which are not engaged by these current anticonvulsant medications. And this suggests a much greater and broader use in refractory patients, which is quite desperately needed. Next slide, Steve. So it might come to us as a surprise to all of you that with all the market and therapeutic potential that CBD has to offer, CBD is actually the furthest thing from a good oral drug candidate. So being a very lipophilic molecule, CBD is virtually insoluble in water and aqueous media, which severely limits its dosage formulations. It also suffers from a very low oral bioavailability of around 8%, and this is mainly due to the significant metabolism that the compound undergoes within the liver. So CBD can also act as a CYP enzyme inhibitor, which if you are taking other prescription medication or even other anticonvulsants, this can lead to off-target effects and a lot of problems. So in order to offset all of these issues, clinicians often prescribe very high doses to elicit an efficacious response. And as you can imagine, this only exacerbates and elevates the risk of these off-target effects and toxicities. So instead of taking the time to solve these problems, GW Pharma basically smothered them in a blanket of sesame seed oil, which is what Epidiolex is formulated in. So it's the same compound, it's CBD still, but just in a sesame seed oil formulation with Epidiolex. So strategy is definitely needed to solve these therapeutic pitfalls and problems. Next slide, Steve. And this is where we come in. So we've developed a prodrug strategy that makes use of both of the phenolic hydroxy handles of CBD with 2 chemically distinct groups, which you can see in the top left corner over there. And both of these chemically distinct groups will serve different functions in overcoming these unfavorable properties I spoke about on the previous slide. So at first, we will have a dye-substituted prodrug shown on the top left, and this dye-substituted prodrug will improve on the aqueous solubility, which will relieve the problems with oral formulation, while also enhancing the metabolic stability of the compound, particularly against gastric fluid and, of course, the very important liver. So once this dye-substituted prodrug has been absorbed, it's gone through the liver and it's circulating within the bloodstream, it's been specifically designed so that group 1 will be enzymatically targeted and cleaved within the blood plasma to liberate the monosubstituted prodrug shown in the middle. And this monosubstituted prodrug that's released will then have its own specific designation to enhance tissue distribution to the central nervous system, while also improving blood-brain barrier penetration. And in doing so, we hope to increase the amount and concentration of CBD at the site of action, which is the CNS, which will obviously lead to a more efficacious response. So a very important aspect of this prodrug strategy is also that at the end of all of this, once the monosubstituted prodrug is within the CNS, it has to release the active parent compound CBD. And if we can do this and show that this is the active component, we'll be able to make use of an expedited approval process following the same avenues that we used for CBD's FDA approval, or the 505(b)(2) pathway, which allows for expedited approval for agents that already have received approval. Next slide, Steve. Right, so we've made a lot of progress in the development of these CBD prodrugs. And I thought the best way to do this would be to just show a comparison of our prodrugs against CBD. So looking at the top left corner, you can see that across the board, CBD's in vitro parameters are very poor, with a solubility of around 17 micromolar, a half-life in liver microsomes of around 6 minutes, and a transport rate of about 0.6 picomole per minute. So we've synthesized over 120 different prodrugs, and we've screened the vast majority of these in a large number of in vitro assays. And in doing this, we've been able to show that our prodrugs reach aqueous solubility of over 1,000 micromolar, which is a 60-fold increase from 17 micromolar for CBD. We've also been able to increase the metabolic stability to over 2.5 hours from 6 minutes, a 25-fold increase. And very importantly, we've been able to show that our prodrugs show far greater brain penetration and transport capabilities when compared to CBD. So this is visually represented in the top right bar graph. So this was an MDCK2 based cell assay, which mimics the conditions at the blood-brain barrier. As you can see, the value in orange over there for CBD, that's the amount of CBD that is transported in these MDCK2 cells, which is far lower than the 4 prodrugs shown in blue on the right of that CBD over there. So that's a great result. Very importantly, we've also been able to show that our prodrugs are converted to CBD both in circulating plasma and within the brain. So looking at the graph on the bottom right, you can see that the amount of prodrug within brain homogenates, shown in blue, steadily decreases over time, over a 6-hour period. And around the 1 hour mark, you can actually see we could track the liberation of CBD from this prodrug, which steadily increased over time. And this is exactly what you want to see with your prodrugs. And we were able to show that over 60% of CBD was released from this prodrug. Next slide, Steve. So the success that we've had in developing these prodrugs thus far can mainly be attributed to the fact that we have access to a large number of these in vitro assays in-house, which was set up by the very talented Dr. Savita Sharma. So we've been able to identify a couple of lead candidates through these in vitro assays, but unfortunately in vitro can only take you so far. And at the moment we're transitioning from in vitro to in vivo assays. And we're doing this in collaboration with Dr. Stephen Trinellis and Dr. Scott Myers. So firstly, we'll be looking at carrying out some PK experiments, looking at the half-life, bioavailability, and tissue distribution of our prodrugs. And once we've looked into this, which is ongoing at the moment, we'll be moving to looking at the efficacy of our prodrugs in seizure models, as well as looking at the safety profile in behavioral models. Next slide, Steve. So just to talk a little bit more about these in vivo efficacy models, we will actually be able to utilize the same seizure models that we use to validate Epidiolex. So this is a Dravet mouse model, and we will be doing this with Dr. Andrea Skeeg and Dr. Jennifer Wong that have a lot of experience in developing these models. And so our goals for this whole in vivo aspect of the project is to firstly identify a lead candidate with a much more reliable PK profile when compared to CBD. And the great thing about this, the fact that we have access to these seizure models, is we can actually do a head-to-head comparison of our prodrugs versus Epidiolex, which will be very telling, as Epidiolex is already an FDA-approved substance. So A very important aspect as well is that we want to see higher brain concentrations of CBD with our prodrugs, as this can most certainly be used as a treatment for this refractory epilepsy patient population. So with higher brain concentrations, you obviously know that it will be a more efficacious drug, so we can use lower and more controlled dosages. So this will lead to a diminished off-target effect profile, which we are also going to be investigating in these experiments. Next slide, Steve. So lastly, just as Nicole mentioned at the end of her talk, this prodrug technology is most certainly a transferable platform. And with the successful carrying out of these in vivo assays, we can show a proof of concept, and we'll be able to apply this technology not only to other cannabinoids such as cannabidiol, cannabidiolic acid, which all show anticonvulsant properties as well. We'll also be able to investigate the other indications of CBD, such as utilization for chronic pain treatment or treatment of anxiety and depression. In fact, this technology can also be extended to other approved therapeutics that also display suboptimal PK and physical chemical profiles, just like CBD. So how does this apply to African scientists? Being a South African myself, I know that Africa really has a wealth of biodiversity and a large base of indigenous knowledge with plants and medicines, and we have great natural product chemists in Africa. So these product strategies that are developed can also be utilized to bioactive natural products that are discovered, within Africa. And the main advantage of this is that while you can't patent a natural product that you discover, you can most certainly generate IP with the synthesizing of novel chemical entities or prodrugs of these natural products. And if you can show that your prodrugs are superior to the natural product, you can most certainly get composition of matter protection, which is very important. So with that, I'm going to pass it on to my colleague, Dr. Stephen Pelly, who's going to talk to you a little bit about the research that they are doing with RSV in the Liotta Research Group.
Thank you, Luke. Yes, indeed. So it's a great privilege for me to be able to tell you about a project that we recently ran in our group. And what I'd like to highlight in this particular project is how we made extensive use of molecular modeling tools to very quickly drill down to highly active compounds in this series without having to make a whole massive array of analogs. All right, so this project's all about designing inhibitors for respiratory syncytial virus, specifically targeting the fusion protein. So just a little bit of background. RSV is well known. It's an orthopneumovirus, and it was discovered in 1956. Annually, it accounts for about 70% of acute respiratory tract infections in infants in their first viral season. And really, it infects nearly all children by the age of 3. So it's not uncommon. The problem is that hospitalization of around 3.2 million children annually occurs, and that's where the problem comes in, and especially since 66,000 of these children may perish annually. Treatment, there's currently no direct acting antivirals for this particular disease, and that is a big problem. So there's a big need for safe and effective therapeutic agents to target RSV. There are a number of targets that people are looking at as far as tackling RSV is concerned, but the target that interested us the most was the fusion protein. Now, the fusion protein is absolutely indispensable for viral attachment. Furthermore, in around 2010, there were a few chemical agents that were actually in clinical development as RSV fusion inhibitors, and they are shown on the screen there. We were particularly interested in the BMS compound, BMS 433771. The reason for this was for a few reasons, actually. First of all, the BMS compound was the only compound that had oral bioavailability, and that was important to us. The other important reason was that development of the BMS compound was actually halted by the company due to a business strategy change. Furthermore, in the process of their development, they had developed a wealth of structure-activity relationship data that we could use in the design of our new compounds. So we decided to make use of this wealth of information, as well as the fact that nobody was pursuing this compound anymore, to see if we could pursue this avenue. I won't go into too much detail about how we did this. Suffice to say that we wanted to drill down very quickly to novel chemical space and have active compounds without synthesizing hundreds of analogs. That's one of the things about academia, of course, is compared to, say, big pharma, is that you don't really have a team of 20 to 30 scientists that you can throw at one project and generate hundreds of analogs. So you need to find a way of quickly getting to active, interesting compounds without synthesizing hundreds of compounds. And to this end, we make extensive use in the Liotta group of various molecular modeling techniques. And our first approach in this particular case was to use essentially an AI learning algorithm, which essentially makes use of a naive Bayesian network approach, incorporating molecular fingerprints. And we launched this from within Pipeline Pilot. In a nutshell, what this algorithm does is we feed it a training set of compounds, and it learns what features on the compounds make for active compounds, what features make for inactive compounds. And then what we can do is we can generate hundreds of thousands of virtual new compounds going into novel chemical space and screen those compounds through the algorithm. And its job is to pick out which of these novel compounds will potentially be active. So this is what we duly did. We screened hundreds of thousands of compounds. And from those hundreds of thousands of compounds, we managed to pick 5 compounds of interest. And these 5 compounds are shown on the right-hand side there, 1 through 5. And we synthesized these compounds. And what's absolutely amazing is the success rate that we had with this particular approach. 4 of those 5 compounds met our criteria of having an IC50 value less than 30 nanomolar. Only one of the compounds was not active as far as in our interest range. So that's an 80% success rate, which is really amazing. Now, at around about the same time, J&J entered the arena with their particular compounds. Now, the J&J compounds are shown on the top left there. And you can see structurally they are quite similar to our compounds and the BMS compounds as far as the overall scaffold is concerned, with just one big difference. If you take a look at the top right-hand compound, on the 5th position of the indole, there's a chloride. There's a bromine on the left-hand structure. So clearly, and I should point out that these compounds were about an order of magnitude more potent than our compounds and the BMS compound. So clearly, this halogen has some important effect. And if you take a look at the structure on the right there, there's very clearly a halogen bond occurring to the carbonyl of the peptide bond of threonine 397. And this is probably accounts for the boost in potency. So we thought, well, we can take our compounds that we've managed to get as far as we have using machine learning and now halogenate these compounds. And maybe we will see the same boost in potency of around an order of magnitude. And so this is what we duly did. We took our most potent compound, compound number 2, which is shown on the bottom left there, and we made these 3 halogen analogs of these particular compounds, the bromide, the chloride, and the fluoride. And if you take a look at the table, you can certainly see that this approach was very successful. Compound number 2 has an EC50 of around 5 nanomolar. By including the bromine, compound 44, we immediately improve our potency by an order of magnitude, going down to 0.5 nanomolar. And this is similarly the case for the chlorine analog 45. What's of interest is that the fluorine analog 46 has a massive drop-off in potency, greater than 250 nanomolar. And this is actually really nice because we can account for this drop-off in potency in terms of the fact that fluorine actually is very poor at forming halogen bonds. And in fact, so now it's not forming the halogen bond, it's just a steric encumbrance, and it interferes with the binding of the compound in the fusion protein. And so just to conclude then, what we've done here is, as I mentioned before, as an academic institution, we don't have the capacity to synthesize hundreds of different compounds. And so we need to use the tools that are available to us to quickly drill down to active compounds so that we can synthesize just a handful of compounds, and those compounds are of interest. Now, having done that, we now have these interesting compounds. What do we do with these compounds? How do we move these compounds or compounds like these in other projects forward, basically getting them from the bench where they're of interest onto the shelf. That is a whole different story, and it is quite complicated. And I'm going to hand over to Professor Liotta now to talk about this in the context of Molnupavir.
Thank you, Stephen. I hope you all agree that I have three outstanding coworkers, and I've had many students and postdocs in my group from Africa over the years, and I've always found them to be very well trained, very hardworking, and very creative. And these three folks are certainly no exception to that. So thank you all for the excellent presentations. So what I'd like to do today is tell you a little bit about what we call the DRIVE model and the development of a new drug for treating COVID-19 infections. As background, after attempting to do drug development at Emory for over 2 decades, I concluded that there were several unintended impediments inherent in the structure of all research universities that made academic drug development more challenging than commercial sector drug development. Now, for clarity, I'm differentiating here between drug discovery, which can work quite well at universities, and the more challenging and complex process, preclinical drug development, which involves progressing a hit, perhaps discovered through a screening campaign, to a clinical candidate that has good market potential, robust intellectual property protection, and a well-defined regulatory path forward. Next slide. Let's examine a few of the attributes of each system. First, commercial sector startups often have seasoned management teams with complementary skill sets, as well as many years of development experience. By contrast, while universities oftentimes have talented scientists, they rarely have management and drug development experience. Second, the management team of the company must be able to execute complex business plans and nimbly respond to problems and opportunities. On the other hand, university scientists usually lack business expertise and quick decision-making capabilities. Third, companies understand the critical importance of developing robust intellectual property protection for their technologies, while universities often seek the least expensive option for filing patent applications. And fourth, while companies routinely reinvest profits to promote growth, universities often distribute technology revenues to the inventors in their schools. This provides little incentives to non-inventors to invest their time and money in the development of the technology. Next slide. In an effort to circumvent these unintended obstacles to academic drug development, 10 years ago, my colleague George Painter and I created Drug Innovation Ventures at Emory, which goes by the acronym DRIVE. DRIVE is a wholly owned, not-for-profit subsidiary of Emory University. However, since it's a separate legal entity, it is not bound by Emory policies or procedures. DRIVE is led by a management team that has discovered and developed multiple drugs. It can enter into fair market value licenses for Emory technology and use its resources and expertise to move licensed technologies to advanced value inflection points. It can also independently negotiate third-party licenses or spin out for-profit startups. DRIVE serves as the commercial arm of the Emory Institute for Drug Development, which uses the acronym EIDD. The EIDD has the staff, equipment, and space required to bring a drug candidate through a phase 2A clinical trial. Finally, since both DRIVE and EIDD are part of Emory, they have ready access to all of the physical and intellectual resources at Emory, unlike typical university startups, which must acquire their own space and equipment. Next slide. Since its inception, the DRIVE EIDD partnership has had multiple commercial successes, but today I only have time to tell you about one of them. Specifically, it's estimated that single-stranded RNA viruses are responsible for 80% of the worldwide viral burden. Yet, with the exception of drugs for hepatitis C, no drugs have been approved to treat any single-stranded RNA virus infections. Several prominent RNA viruses are shown on this slide. Prior to 2019, my colleague, Dr. George Painter, who partnered with me years ago on the development of emtricitabine, had identified EIDD 1931 as a broadly active inhibitor of multiple single-stranded RNA viruses. In early 2020, he learned through collaboration with colleagues at other universities that EIDD 1931 was active against the SARS-CoV-2 virus. Next slide. After further investigation, it was determined that while active, EIDD 1931 had poor permeability, which limited its ability to be used as a drug. However, by converting it into a 5' isobutyrate ester prodrug called EIDD 2801, which is you'll see on the left, the permeability was significantly improved. Once absorbed, the prodrug is sequentially cleaved by endogenous esterases, and the resulting 1931 compound is anabolized by endogenous kinases to its corresponding triphosphate, which is in turn incorporated into a growing strand of viral RNA. Although many viral polymerase inhibitors function by chain termination mechanisms, incorporation of this compound actually allows RNA elongation to proceed. Viral ablation occurs by a different mechanism that I'll show you on the next slide. One of the common features of antiviral therapy is that single agent treatments, which we call monotherapies, almost always lead to the emergence of resistant mutants, which then rendered the drug useless for further treatments. However, one of the unique features of EIDD 2801 is that it can be used as a monotherapy. This is because the oxime functionality can readily tautomerize to generate both a uridine-like and cytidine-like nucleotide, which can base pair with either adenosine or guanosine, respectively. Over time, this results in an accumulation of a catastrophic array of mutations that ultimately lead to the death of the infected cell. Next slide. To finish up, I'd like to summarize the current development status of EIDD 2801, which is now called Molnupavir. Molnupavir is a direct-acting antiviral agent that is orally bioavailable. It can be administered as a capsule or in an aqueous solution, and importantly, can be prescribed to symptomatic patients who won't need to be hospitalized to take it. Since most of our hospitals are operating at full capacity during the COVID-19 pandemic, this drug could, over time, significantly contribute to reducing the number of COVID-19 patients being hospitalized. In addition, Molnupavir can be used prophylactically with individuals who are at high risk for getting infected, much like we do with PrEP for HIV. Drive has entered into a license with Ridgeback Biotherapeutics for the development of Molnupavir for treating SARS-CoV-2 infection. After completing successful phase 1 clinical trial, Ridgeback subsequently sublicensed Molnupavir to Merck. To date, a phase 1 single and multiple ascending dose clinical trial and a phase 2 clinical trial have been completed. Phase 2-3 studies are underway internationally. Emergency use authorization has been applied for in India, and importantly, the US government has made a $1.2 billion purchase commitment for 1.7 million courses of Molnupavir, and the actual regimen consists of taking 10 pills, 2 a day, and usually at the end of 5 days, the viral load is undetectable. Finally, Molnupavir will be one of the drugs supported by a $3.2 billion US government drug development program. So Molnupavir has the distinct advantage over other COVID-19 drugs by being orally bioavailable. And as a consequence, it has become the leading direct-acting antiviral drug candidate for the treatment of COVID-19 outside of the hospital. With that, next slide. With that, we'd be happy to answer any questions you have. Thank you very much. I don't know how we've been doing Q&A in the conference, but one simple way would be to type your question into the chat box. Here's one. Just a nice comment. Thank you. Well, if there are no questions, perhaps we should then just end the session. And I'd like to again take the opportunity to thank you all for listening. Oh, well, there is one. Sorry. How would you suggest African academics tackle the challenge of drug discovery on the continent? So this is an interesting challenge because drug discovery is a very competitive field. Luke alluded to the fact that Africa possesses a huge amount of biodiversity, many natural products, and the problem with natural products is that, of course, many of them are complex, and many of them can only be isolated in small quantities. But once you find something interesting, the problem with them is that you can only patent use of the compounds for treating individual diseases. So the opportunity to modify them with prodrugs specifically, as Luke pointed out, is a really interesting one, because if you can show superiority of your prodrug modified natural product relative to the natural product, then you should be able to get composition of matter protection for the compound, which is really a game changer. So one opportunity, and if you do other modifications that alter the core structure, then it's a different compound. And of course, you can take that route as well. But if you know the natural product works well, but it just has a few defects in it in terms of, let's say, its pharmacokinetics or other things, that's one strategy for going forward. But I guess I would advise African scientists, because of this wealth of biodiversity, that would be an excellent starting point for you to pursue drug discovery in ways that others couldn't easily do. All right, well, thank you all very much, and we'll be signing off. I hope you enjoy the rest of the conference.